Well, happy Friday, everybody. Hopefully, all of you are staying safe and warm. Got a little bit of snow out there today, so stay inside, get your hot cocoa, fix yourself some espresso, kind of get a double shot with a little bit of steamed milk on top. Call it a latte. All right. So, need to buckle down and get some things done today, people. All right. So, here's what you're going to be doing today. As you know, over in the lab the last couple of days, we've been working on something known as protein synthesis, and that's basically where you take the instructions that are located in DNA, the sequence of A's, T's, G's, and C's. That tells you what protein you're going to make. All right. So what we're doing now is trying to figure out how we can get from point A to point B. All right. So the first thing we've got to do is copy the DNA inside the nucleus, and that is called transcription. That's where we take messenger RNA and we line it up with the DNA. All right. And then the messenger RNA actually leaves the nucleus out through those little nuclear pores. Okay. So now we've got the messenger RNA out inside the cytoplasm. Where does it go? It goes to a ribosome. Okay, a ribosome is where the proteins are actually going to be assembled. In order to assemble proteins, we've got to have amino acids, right? And so basically what happens is, now again, we're out in the cytoplasm at the location of the ribosome. So we've got to get some amino acids. Those are the building blocks of proteins. In order to build proteins, you got to have amino acids, okay? And so transfer RNA, which we call tRNA, brings the appropriate amino acid to the site of translation, okay? So we had transcription was the copying of the DNA. Now we're translating it into figuring out what protein we can actually make. All right. And so translation takes place in the cytoplasm at the ribosome where tRNA is bringing the amino acids in, and it will bond them all together. All those amino acids get bonded together to create a protein. Then that protein folds up into a three-dimensional structure, and it actually does something. It has a function for the cell. All right. So our goal today is to review that process. Okay, the transcription of the DNA into mRNA, messenger RNA, and then the translating of the mRNA out in the cytoplasm into making proteins. All right, so you're going to be doing a review activity for that. All right, this is pretty important stuff. All right, this is how organisms are made. All organisms are made out of proteins, and so we've got to figure out how to build proteins. All right. So follow the directions below. Have a safe weekend, and we will see you on Monday.